Well, I appreciate the opportunity to present Orthocyte to you today, especially the placement in the agenda right after, towards the end of the first day, right before cocktail hours. I was expecting full attendance today, which is good. Just from a disclosure perspective, Orthocyte is definitely not a clinical stage company. We're very much a preclinical stage company, as you'll see. So as a result, I'll present to you not what we have done, like you've heard a lot today, stories of how we were successful or where the challenges were. But I joined the company about 6 months ago, so I'm going to present mostly our plan and how we actually plan on staying alive and actually making a little bit of money and hopefully reach the blockbuster stage. So this is how I structure my presentation. I'm going to tell you a little bit about the opportunity, the unmet need in orthopedics that we're facing, the challenges, our technology, and how we can address this unmet need, and finally our strategy and how we're tailoring our approach to meet that challenge. So Orthocyte is a member as a part of the Biotech family, and I'm not going to talk about Biotech. That's a whole separate 2-hour Mike West presentation I'm sure some of you might have heard before. But Orthocyte is the subsidiary that deals with orthopedic, and as you know, orthopedic is the specialty which deals with bones and whatever is attached to bones, so tendon, ligament, and so on. And our therapeutic approach is regenerative medicine, so which, as you know, is any kind of combination between cells, scaffolds, and bioactive. And you'll see that we actually have position in all of those. So orthopedic is a little bit different, as you might expect. It's from a health economics perspective. It's different than other unmet need in that it's not a life-threatening condition most of the time. It's not like losing your cognitive abilities in neurodegenerative diseases. So it's a little bit different challenge in terms of health economics. But the difference is that it affects nearly everybody. So whether it's affecting whether you're talking about knee pain stemming from cartilage injuries, or, as you'll hear more today, the intervertebral disc damage that leads eventually to chronic back pain, it's very, very different. They affect nearly everybody. But the pathophysiology is very similar. In both cases, these tissues are very avascular, and it's very difficult to reach therapeutic dose for any types of pharmaceutical-type drugs because these tissues are almost completely deprived of blood vessels. And the problem is, once you have an injury to these tissues, they don't tend to heal themselves, and it's mostly because they're avascular. And they involve both soft tissue and bone. So the problem is, once you have an injury to these tissue, eventually they degrade, and there's really no treatment option between the initial injury, for the most part, to when you actually need invasive surgery. So looking at back pain specifically, which is the focus of Orthocyte right now, it's a huge health economic burden. The reason for this is because it actually affects the physically active population. So it's not just a disease of the aging population. It actually affects everybody in their productive life. So between 18 and 65 years old, it's nearly 75% of the people actually are suffering from chronic back pain. So actually, it has a big impact on medical costs, as you can imagine. So if you want to understand how you can address this disease of chronic back pain and disc degeneration, you have to understand how it actually happens. And this is what's happening the disc itself is the structure between the vertebral body. It's what allows the spine to bend and absorb shocks and protect your spine. So once you have an initial injury to this disc, over time it starts degenerating, and there's initially an insult and an inflammatory reaction, which doesn't go away, which doesn't go away. It leads eventually to matrix breakdown and cell and tissue loss, and eventually the whole tissue collapses, and this is when you actually need surgery. So it's a very slow and progressive disease with many opportunities to intervene. Like I said earlier, between pain medication in the early stage of the disease, and when you're hurting so much that you need surgical intervention, there's a really huge therapeutic gap when there's really nothing that actually works or can slow down or reverse this disease process. So there are several ways of intervening. Depending on the mechanisms of action, you can intervene potentially with stopping this inflammatory response initially, or stopping the matrix breakdown or cell loss, changing the catabolic environment to more anabolic, and eventually replacing the cells when it's a later stage disease. Like I said earlier, the disc itself is the largest avascular structure in your body. So it's very, very difficult to reach therapeutic dose in this environment. But what it means, though, is from a therapeutic approach perspective, it's actually a really interesting one for regenerative medicine because this tissue provides us a unique environment in that not only, so you can actually potentially consider an allogenic approach where the cells could potentially be, would potentially allow to survive a little longer than, say, other vascularized tissue. So it provides an environment that may be more immune privileged than, say, other tissue, which for us is an advantage. So regenerative medicine is an actual, it's a real opportunity, it's a real market. In regenerative medicine, there are actual products in this space. It's not just a market opportunity, it's actually a market size. If you just look at bone fracture or spinal fusion and considering spine alone, there are today just ceramic-based product or graft product, which are very low potency, it accounts for about $500 million in sales today worldwide. And if you look at recombinant growth factor in this space, it was back in 2010, it was almost $1 billion. So end-stage product together, we're already $1 billion market today. So if you look at potentially treating the disc, treating back pain earlier in the disease process, where they are far more patient, at least 10 times more patient, it's easy to figure that you can potentially reach the multi-billion-dollar market opportunity, whether you're extremely conservative or very aggressive in your projections. So our technology, we have developed at Biotech, as some of you might have known, a unique technology that's based on embryonic stem cell technology. But what is different is that although we started from embryonic stem cells, the cells were actually induced to differentiate using multiple differentiation protocols. And what happened is we isolated cells, clonally derived cells, in the different, this doesn't really work anymore, but anyway, so we isolated cells in the different branching point of this differentiation tree. And we can actually cover the entire body using cells that can potentially repair all of the tissues in the body. So the cells were isolated at clonal stage and propagated and characterized. And what I hope to convince over the next couple of slides is that we have enhanced attributes as a result. So we have cells that have the broad applicability of embryonic stem cells, but have the targeted utility of mesenchymal stem cells. So we have enhanced attribute in terms of diversity, purity, identity, potency, and scalability. So in terms of diversity, we have isolated over 200 diverse lines, all different from one another. And this just represents a heat map of 200 lines you can see horizontally, and about a few hundred different genes that were screened. So we have extensive microarray data, pre and post differentiation. And you can see that these lines are, some of these lines are grouped in families, but still they're very different from one another. So they have similar characteristics, but they're all diverse. In terms of purity, as I said before, they're clonally derived, which means that they're extremely reproducible in terms of redifferentiation and and their behavior, both in vitro, and in vivo. So they're far more easier to control, say, than the original embryonic stem cell, because they're already committed to a certain pathway. in terms of identity, like I said before, we have extensive microarray data, so we know extensively the expression profiles of all of the clones pre and post differentiation and along various differentiation induction protocols as well. So in terms of potency, and this is really why I joined a company. I come from a musculoskeletal background, and when Mike West showed me this data, this is when I became really interested in the company. This is just an example of 7 chondroprogenitors. So these cells have the ability, these 7 different clones have the ability of forming cartilage in vitro and in vivo. But they express various levels of type 2 collagen and type 10 and COMP and other markers of cartilage differentiation. But what they all have different profiles. So they express these markers at different levels, which is really exciting because now we can potentially select the attributes that we need for various indications. For example, a articular cartilage therapy might require a specific cell type that is well adapted for that environment, whereas a bone cell or a ligament cell might be better, in different conditions. And this is what I mean by this is just an example of where you have MSCs on the left side, which will do what MSCs are more or less programmed to do in a specific environment. And in our case, we can select, and this is just an example of these 7 lines. We have now over 40 osteochondral progenitor that we can actually select attributes from different perspectives. So we have chondrogenic and osteogenic cells. That is, these cells can become cartilage or become bone, but also they have various level of bone-inducing and cartilage-inducing factor, for example. So they have the expressed growth factors that are specific for, or characteristic, should I say, of bone induction, so, such as BMP or bone morphogenetic protein, or GDF5, which is more of a chondrogenic type of growth-inducing factor. So we can select, or we can pick cells that have various characteristics based on our needs. We can look also at they express various level of matrix degrading or matrix inhibitors, matrix degrading inhibitors, or antagonists. We can select based on that, as well as cytokine modulation. So there's various characteristics that we can select. So we no longer have to force fit a round peg in a square hole. So we can actually select the cell that fits better the requirement. From a scalability perspective, this is just a population doubling study, and you see MSCs at the bottom there, at least in our hands. And our lines, a lot of our lines can go to well over 100 population doubling and actually have a very stable phenotype under these conditions. So at 40 to 50 population doubling, we know we can generate over 100 million doses of about 100 million cells and retain the specific phenotype. The second half of our, or the second piece of our technology is this high-stem hydrogel. We have a biomaterials platform. And what's interesting about it is it's derived from hyaluronic acid and collagen, which are the natural component of a lot of connective tissue matrix, as you know. So it's functionalized hyaluronic acid and functionalized collagen, which we can mix with a cross-linker. And what's unique about it is it's very cytocompatible. So it's very good at it's very compatible mixing with cell. Sorry, and we can actually preserve cell viability while the gel goes from a liquid. We can mix the cells in a liquid format prior to implantation, and then adding the cross-linker, we can actually deliver in situ and form a gel implant that contains the cells while maintaining viability and not compromising the activity of these cells. But we can also mix anything we want, obviously. We can mix in ceramics or other type of materials that are less with less regulatory burden and have opportunity of developing more near-term types of products. So our strategy. So as I told you, we have numerous tools for addressing numerous musculoskeletal indications. But if we had the choice to make at some point. If you look at, we have cells that can form cartilage or disc tissue. So if you look at the development pathway of whether you look at cartilage injury or disc injury for back pain, and if you look at just the preclinical requirement between the two pathways, and you know that cartilage injury requires large animal model of 6 to 12 months at least of follow-up preclinical, whereas in back pain or degenerative disc disease, we know that we can get away with 3-month follow-up using small animals such as rabbit. And likewise in clinical development, as you know, as you'll see, and you have seen perhaps already, it requires at least a year, most likely 2 years of follow-up, for cartilage injury. Whereas in pain or degenerative disc disease, where we look at mostly pain and function, a 6-month follow-up is enough. If you look at the addressable pool of patient, which is far bigger in chronic back pain or degenerative disc disease, and the health economic benefit that you need to demonstrate beyond the approval, and again, for cartilage repair, you need to have this sustained repair that requires you at least 3 to 5 years, and where back pain, it's just a different value proposition. So all in all, if you compare cartilage injury or cartilage repair, say, with degenerative disc disease, you can actually save, both in the preclinical and your pilot clinical study and your pivotal study, at least a good 4 years, which allows you to really ramp up revenues in the early stage, or in the late stage, I should say. So this is what we did. As I told you, there's a huge opportunity between, say, Tylenol or Motrin and invasive surgery and this slow degenerative process. But what we need, the therapeutic targets, is we need to form disc or repair disc in the early stage of the disease, and we need to form bone in the late stage of the disease, because when your disc is completely degraded in stage 5, the only thing you can do is really fuse it to minimize motion. So our product development our strategy consists in this. We have this biomaterials platform, and like I said, we can combine not only living cells, but we can also combine it with these ceramics or osteoconductive materials and form bone graft material. And these types of material, they're 510K cleared, they're still regenerative medicine, they're obviously very low potency, and they're commodity. There's tons of these products on the market today in this $500 million segment. But there's a very clear precedent in developing these types of products, and this is what we intend to do, or how we want to utilize our platform initially. And then the second generation is we'll combine the same type of biomaterial. Now we're going to enhance them with our pure stem line, either for example, to form or enhance bone for spinal fusion, or use our disc cells or cartilage cell to repair discs. So this way we have a balance of near-term and long-term product development strategy. So this is the therapeutic approach for us. As I said, based on our pure stem, we have different mechanisms of action that are possible based on attribute that we can select ahead of time. We have cells that could potentially slow down degradation, that are expressing high level of matrix degradation inhibitors. We have cells that are chondrogenic, that is, they can perhaps induce the residing cells to switch from a catabolic to an anabolic type of environment. And then we have cells that are discogenic or chondrogenic themselves, that is, they could potentially be used to replace the lost cells. And at the end, at later stage, when the only thing that can be done is actually induce bone, we have cells that are osteoinductive as well. So for Orthocyte, from my own perspective, I don't really care in a way. I don't want to say this too casually, but I really don't care which mechanism of action is going to ultimately work. To me, what's great about our opportunity is that we can select the one that will actually, we can screen the one that will actually work and pick the one that's going to be the winner. So this way we can intervene not only in the late stage of the disease, but much earlier stage where there's a lot larger patient population with a more diversified set of provider, address a broader set of provider, and provide a much bigger opportunity for And trust me, just the end-stage disease, like I said earlier, is still $1 billion market today. So obviously addressing a larger population really represents enhancing this market opportunity. So in a nutshell, this is what we're doing. We're in terms of balancing and sharing this risk is, like I said earlier, if you look at on the top of this slide, we want to leverage our biomaterials platform and develop these bone grafting material that, like I said, they're still regenerative medicine because they still will be turned over and replaced by normal bone, and they're used to enhance bone regeneration. And meanwhile, while we're launching these products that can be developed, that will be developed in the next 12 to 24 months, and in several different configurations, there's really an opportunity of launching several products that will start generating near-term revenues. Meanwhile, what we do is continue the development proof concept of our more blockbuster type of approach in terms of using a cell therapy type program for bone induction and disc repair. And while we're going through IND enabling and then pilot and pivotal clinical, we can raise valuation and with the near-term product development, balancing this, perhaps delaying or not going to investors as often as traditionally was done. So, in a nutshell, what I want to leave you with is that, Orthocyte specifically has this balanced, near-term revenue-generating product pipeline. So we're not only placing several long-term bets in several musculoskeletal indications, but we're really balancing this with near-term product development, which hopefully will allow us to place ourselves favorably with future investor and partnering opportunities. Thank you.
Thank you very much.